now you can start to combine, say, biotechnology with computer programming or AI to try and then get into this field, which I think it's going to be huge, right? If you start thinking about you keep hearing this on the news, and I've been hearing it for years and years when I was at Sick Kids Hospital, individualized medicine.
That's right.
Right?
Just hasn't happened yet.
No, it has, but we're on the cusp, and I think it's going to happen in my lifetime. We're right there, right? And now with the advent of AI, individualized medicine is going to be the future.
The following conversation is with Dr. Gregory Anderson, one of the founding professors of the Biotechnology program and a staunch advocate for research at Georgian College. He joined the faculty in 2015 after spending 12 years as a researcher at both Sunnybrook and Sick Kids Hospitals in Toronto. While there, his research focused on using high-resolution, high-throughput physics imaging techniques to try to understand the roles that various genes played in the development of the cardiovascular system and ultimately their contribution to cardiovascular disease. This work required the collaboration of various scientific teams from around the world, which helped foster in Dr. Anderson the idea that truly great research cannot be done without truly great teamwork skills. Dr. Anderson holds both an MSc and a PhD degree from the University of Toronto in medical biophysics, as well as a BSc from the University of Waterloo in biology and chemistry. Dr. Anderson is well versed in the scientific method, and he's published 13 peer-reviewed papers on his research, most recently in 2020. Today, Dr. Anderson continues to contribute to research at Georgian College as the research lead for health and wellness sciences. And without further ado, Dr. Gregory Anderson. Thanks for joining me, Greg.
No problem. I'm very happy to be here.
What I wanted to talk to you about today is a little bit about your research experience and your time spent in research, because for a lot of students, when they hear about research, or even lay people outside of the research world, they have an impression of it that comes from TV or movies, that sort of thing. So I wanted to talk a little bit about that. CSI, exactly. So tell us, before we get into what it's like, what motivated you to get into research?
Great question. So when I was in undergrad, I actually was in a co-op program. So I went to the University of Waterloo, which is very well known for their co-op program. And so I was lucky that way because, I really didn't know what I wanted to do. My first couple of co-op terms were more in industry. So I was working, I worked in, it was all chemistry related, but it was all in like a plastics factory and then a foam factory. And it was great, but it was industrial research. But then my last co-op terms were actually two, because it was two in a row. I was down at the University of Notre Dame doing research there, more, the type of research that's done in a university. And so that's kind of where I fell in love with the whole idea of academia. And I had a really great supervisor there who, was a fantastic scientist, and we were doing some really kind of cutting edge, it was kind of chemical physics research. So looking into carbon nanotubes and how we can use that for solar energy conversion. Right. We had to do things on a small scale before you could ever ramp it up to, say, solar panels. And so that's kind of where I fell in love with it, because it was very much the way that the research world works, not the industrial side, but the kind of academia side. And so that was right before I started my fourth year, my last year of university. And so that's when I started seriously thinking about my next steps, because you inevitably have to at that point too. And so I asked myself, where do I want to go? What do I want to do? And you don't really know necessarily, right? You're still pretty naive, I would say, in terms of, I knew what I liked, but you don't know how much research opportunities are out there. But then I applied to a few schools, and the one that I fell in love with was the University of Toronto, and luckily I got in, and then the rest was kind of history at that point. Yeah.
Now that's a similar story for a lot of people who do their PhDs or research now, and undergrad students and undergrad students and college students alike. A lot of them come to a point where they don't know what to do, and they consider research. Now, you were interested, genuinely interested in it. Would you advise against doing research just because, when you're not necessarily wholeheartedly into it, as a stopgap measure? Because some people do that, and it doesn't work out well.
I agree, and I saw that happen a lot. I definitely saw that happen a lot, but sometimes you don't know if you're going to like it. It's a very different life. I know that you know this, but going into a heavy research field is not a 9-to-5 job.
So tell us what it's like for people on the
Okay.
outside. What is a research 9 to 5, or rather a research day or week or month or even a year look like?
Yeah, so when you start in any kind of lab, it doesn't matter what level you're at or anything like that, you have to kind of define a project, right? Usually that laboratory is working on umbrella topics, right? So within that umbrella, there's lots of avenues to go down. And so the first step always, right, which maybe is even in some ways the hardest part, is trying to define your own niche, right? You're doing new things now. You're not an undergrad student where you're learning everything that's already been done. Now it's like, okay, this is what's been done. What can you do to take it even just that tiny step further, right? So defining a project. So I'd say, like I said, it's not 9 to 5 in the sense that depending on the types of experiments you start running, it kind of decides how many hours you're going to be there. I didn't know many people who would be working less than 60 hours a week. I would say if you really want to do things properly and kind of
And if I had to guess, you probably slept a couple of times.
I did, yeah.
Yeah,
So one of the issues for me, not issues, but things that came up, right, is that I was working with mice. And so you have to do certain things at certain times without getting into specifics when it comes to research with mice and working with cell lines, right? I would always go in on weekends to take care of cell lines because you can't let them overgrow. Sometimes when you're checking stages of reproduction in mice, you sometimes have to go in at midnight because everything's on 12-hour blocks, right? So I'd go in at midnight to check on my mice, sometimes on a weekend as well. So there's no... The work week is just do as much as you can, essentially, right? You do need to find ways to not make that your entire life, otherwise you'd probably go crazy. But I would say, like in that type of research-based world, I don't think I ever worked less than 60 hours a week. So it's not for everyone, that's tough.
So even though research itself seems to be, using the scientific method, it seems to be very regimented. Oftentimes it's very not regimented. You're coming in at midnight when you don't expect to or don't want to. Right. You're staying later or coming in much earlier. So even though you might think it would attract people who are very disciplined, I don't know about you personally, I met a lot of eccentric people in the research world because that lifestyle kind of,
I knew lots of people who only worked at night and not during the day, things like that, because you essentially make your own schedule, right? Because at the end of the day, it's not a job where it's like you need to have this done, in the next day or two, which is sometimes actually the most difficult part, right?
Yes.
If I'm working now and I know I have to teach a class on Wednesday, that's a hard deadline. I have to have everything ready by then. Whereas this is much more open-ended, right? I would meet with, say, my committee, my supervisor, and a group of other doctors in the hospital, like once every 6 months or once every year, and they're just asking on your progress, and you want to have made progress, but there's no one looking over your shoulder, so it's really up to you to be. You have to be very independent. I think that's the number one thing I would say is that
100%
independent work. Sorry, that's my cat running around. Very independent work, and so you have to have an independent and critical thinking. You have to have independent critical thinking skills.
Yes, that's right.
Because you're on your own. You collaborate, with people, but it's your project, right? So sometimes things overlap with some other people within the lab. And I was in a really big lab. Of course, and equipment and things like that. But at the end of the day, this is your work, and you either get it done or you don't. And honestly, if things fail more often than they go right.
For sure. I think that's one thing that people on the outside probably don't realize or appreciate, how often things fail. How often you have to repeat an experiment.
It's
not to force numbers results, but because simply something failed.
Something
and you
fails.
Have to do it again.
Yeah, inevitably, like I said, more often than not. And something I tell my students all the time, right, is that I would actually argue that you learn more from the things that went wrong than when it went right. And frankly, this often changes the course of where your experiment's going, right? So even when you start talking very basically about the whole scientific, the whole scientific theory and the steps you're supposed to take, right? It seems like there's 4 steps, observations, experiment, conclusion. I always say, like, your hypothesis, whenever you come up with any hypothesis, one of the main tenets, right, is that it has to have the ability to be proven wrong, and often it will, and that's not bad. That's not a bad thing.
Absolutely not.
Like, that happens all the time, because then you're like, oh, that wasn't what I maybe thought or expected, but this, okay, now I've got to follow this path, and then you start course correcting through all of the experiments. It could be minute things, like, I had to do a bit of coding, not much, thankfully, because that's not an area I enjoy, but I had to do a bit of coding, and sometimes it'd be something, I didn't do that one little step there. Okay, I better change that. It didn't help. Or it could be something massive, right? Because sometimes I would have, for instance, experiments that were months long, and then it fails at the end, and you feel like, oh my God, I just wasted all the time. But it's actually, you have to get out of that mentality of that was a waste.
Yes.
Because it's actually like, okay, now I've learned that.
This is a sunk cost fallacy. You've sunk in so much cost into it. You don't want to start from scratch, but oftentimes it's the best thing that you can do.
Absolutely. And it can be very demoralizing sometimes. But again, you have to change the mindset and go back and say, okay, what did I learn? I mean, you could apply this to life in general, right? But what did I learn? What do I have to do next time? Or what's another possible option for next time that might also fail?
Something that you mentioned before was working with mice and coming in at certain times because that's the way mice work. I think this is something that's not appreciated about biology as opposed to chemistry, for example. You're dealing with living things that have their own timeline. They have a metabolism, they have to live, they have to grow, and oftentimes you're setting up an experiment for a week before you can even execute it because you're waiting for cells to get to a certain density or mice to get to a certain age, right?
Yeah, absolutely, right? So sometimes I would have towards the end, especially a lot of the projects I was working on, I had 3 different mouse strains that I was taking care of, right, for this one particular project. Mice do what they want to do. They don't listen to me. Like when I need them to say reproduce, they might not do it on the schedule that I want to, and then I've got things going at different times, which is really, really difficult to keep track of. But biology is not... I think this is the problem that comes out of undergrad a lot, right? Is that you're used to these experiments you do in the lab that are very regimented. You've got 3 hours, whatever, to do it, and that's enough time, right? But the real world is not like that at all. It's free flowing, and you've got to, I guess, roll with the punches is a way of saying it. But you just have to
be able to adapt.
You have to be able to
resourceful.
adapt. Yeah,
is I think one of the very important skill for research.
100%.
So let's talk about one of your research projects where you were doing targeted mutations of proteins in the heart to look at heart development and also the development of heart disease. So tell us, what is the concept behind targeted mutation?
Yeah, so the basic idea, right, is to always... It never comes out of nowhere. You don't just randomly select some sort of a gene. So in most cases, I would say, in every case I ever saw anyway, there's some sort of human disease, right? That maybe somehow is tracked back to one possible gene. I mean, would be the best case scenario if it's only one gene. That's actually fairly rare. Or there's some sort of small thing that has happened and it's like, okay, this gene might be responsible for this. So let's take a look at it, right? So there's some sort of problem in the human population. So for my project, for instance, there was this, or is this disease called HHT, which stands for hereditary hemorrhagic telangiectasia. And so they propagated this and did sequencing, and sequencing took a long time back then to discover it was this one gene.
Sequencing of people who had this HHT and it's correlated with this mutation.
And they had this mutation that caused this one problem. Mutation in this gene leads to these downstream consequences. So the idea then was to then go, and this was done before I got there, right? So go back to the mouse genome now, because a mouse is a mammal. Development is on a different time frame, but the exact same, essentially. Find that gene, right, in the genome. It's on a different chromosome in mice, but nonetheless, discovered where that chromosome was. And so the idea then is to, using various molecular biology techniques, you do this in cells, right, in different types of embryonic stem cells first. You find a way to essentially corrupt that gene or just delete it. You're deleting it, for lack of a better term, right? You're kind of taking it out.
Yeah.
And you have to have what are called reporters on the other side so that you can make sure that it went into the right place. Now, once you've grown these things in cells.
And were you doing this by introducing stop codons early on in the transcript?
Yeah, 100%. Okay. So we would put a stop, or what they, again, they did this before I got to the lab, but stop codon, put where that gene is. It's on just one of the, just one of the strands of DNA so that you could actually get viable pups and everything. So now introduce it into what's called a chimeric mouse. This is a big process, but I'll kind of keep it in a nutshell here, into a chimeric mouse. That chimeric mouse can then give birth to pups that either have are no mutations, one mutation, or both genes have been mutated.
Right.
And then we can study what are, the heterozygotes, they're called, the ones that have one mutation, and the homozygotes on either side that have either 0 or 2. And then see, okay, so this problem is this is a problem in humans. What is actually happening in those really early stages of reproduction, right, or of gestation, of development? And so in this case, it was something, this is where there was debate. A lot of labs that were studying this thought that it was a problem with blood vessel development, right? Whereas others were saying, no, it looks like it's more of a heart development problem, but these two things are obviously completely intertwined. So it's really difficult to say which comes first, right? So that was the whole essence of trying to discover the problem. So yeah, targeting mutation to delete or essentially corrupt a gene in the same way that it is mutated.
Truncate the protein to a point where it's useless.
Yeah, exactly.
So before you got there, you had some of your colleagues identify a gene that was correlated with this disease, and so you decided to study it, you introduced a mutation, and now you're at the point where you have these chimeric pups, or pups from a chimeric mouse. How are you measuring the outcome of this mutation at the end of the day? What was your output?
So it started from more of a biology standpoint, I would say. So obviously what we found, right, because this is when I came in, they had the mouse line, was only just beginning, right? So all the molecular biology stuff had been done. So the mouse line was up and running. And so then it's like, okay, we need to study early development. So you're looking at embryos. So what we found, right, is that in embryos that had double mutation, they wouldn't survive at all.
Right.
So you wouldn't see this in the human population, right? But it's actually almost the more interesting story, because it really said, this is a really important gene somehow, because without it, if you have 0 of this protein being made, death, right? Unfortunately. Heterozygotes where only one copy is mutated were being born and inevitably looked, you don't like to say normal in a genetic sense, but looked the same as their homozygote counterparts. So the initial steps were always to do kind of classic biology techniques, histology, right? Looking at is there anything different? If we slice through the heart or different blood vessels, are things enlarged? Are they too small? Are they developing at the same speed compared to, again, the homozygote with, no mutations? So a lot of that was done at first.
So microscopy with slides.
Very microscopy heavy, and looking at the stem cells themselves that you can then get from these, see how they grow differently, and like a lot of in vitro work, right? That's usually how you start these types of things, is a lot of in vitro work.
Right.
And so we would look at, okay, if we have these stem cells, what do they grow into? Do they form those really initial blood vessels that you can actually get them to grow in a petri dish? And we even found that the heterozygotes were not perfect, but they were still getting to the point of being born, but then there would be downstream consequences similar to what we saw in humans.
So you were harvesting stem cells from these mice that you generated, and then tried to grow them in culture and see what happened. Yeah,
exactly. So that was those were definitely the initial steps, and then.
So at the end of the day, for most researchers, they're never there long enough to see the end of a project, or that's because they're not there. Essentially, the projects last for the lifetime of a PI, a principal investigator. So your boss is a PI, and their expertise is something, and they work on that their whole life. So a project doesn't really ever come to a conclusion for the most part.
Yeah.
But with the data that you gathered, what was your hope in terms of what would come out of that knowledge?
Yeah,
long
so
term?
so from there, right after all of these more traditional biology techniques, that's when I switched over and started doing more imaging physics work. So the idea
Same lab,
was,
same project?
nope, new lab now
Okay.
that was much more imaging based.
Okay.
And so I was using a technology called optical projection tomography. So now we knew this biology, the biology side. I mean, so it's still asking biological questions, but basically imaging these things in a three-dimensional manner, right? Just using optics, light, with various fluorescent antibodies to highlight certain areas, right? To then create three-dimensional digital maps of everything, so that now you can really start to quantify things, right? I'm not going to say the problem, but one of the issues with a lot of traditional biological techniques like histology is that it's very qualitative.
And subjective.
Right? Yeah, and subjective,
Right.
and there's inevitably errors with histology, and
Of course,
you can
yeah.
make false conclusions. But now that we're not actually cutting tissue anymore, we've got maps, three-dimensional maps, and you can picture this on a computer where you can zoom in and you can color code things and actually get like volume data and put numbers on things and say this
Right.
left ventricle is 0.25 millimeters cubed bigger than it should be.
Right, right.
0.25 is actually too big a number, but you know what I mean? Like 0.0025 millimeters cubed too large at this stage of development compared to what should normally happen, right? So that was where it was kind of going to the next level, like quantifying developmental biology. And so that was
Right.
always my hope. And so then we were studying other mutations as well that were similar but different, to try and compare things, because now when you have numbers, you can compare different types of, say, genetic mutations that affect similar parts of the body.
Right.
Hopefully leading up to this bigger idea of like systems biology, right? How do we start looking at the
whole
totality?
system.
Yeah.
Now, what's really cool about the technique that you were talking about using antibodies for this imaging is that you're essentially using molecular biology to look at things from a 30,000 foot level on a morphological
Yep.
level, which is very, very cool. So the question for you is, what is one of your favorite techniques or technologies that you used in terms of biotechnology during your time in the lab?
So this is why I have a big love for the field of optics, right? So I always say I'm a biophysicist, but the physics side of me is all optics based. And so I just, at the time, 15, 20 years ago now, this technique, OPT, optical projection tomography, was brand new. Basically, the reason why it came about, which I've always thought was a great story, was that physicists said it couldn't be done. Light can't shine through tissue, it gets scattered, you won't get any data,
Right. Any
can't be
usable
done.
data from it, right?
And there was a biologist in Scotland who said, I'm just going to try it anyway, right? It's almost like that classic story of a bumblebee shouldn't be able to fly, but it doesn't know that it's not aerodynamic, so it does it anyway.
Yes, exactly.
And so he was like, I'm going to try this anyway. And so he created this system. Now you have to go through a lot of what are called clearing techniques to actually avoid these issues of light scattering when they hit tissue.
Right.
But he developed a way to do this. And then all of a sudden we can see through tissue when we have different contrasts, and then we can add antibodies and we can light up different tissue types. And so that was that was the whole crux of my whole doctorate degree, was using this technique and advancing it, because it was brand new. And we even created our own system. We improved upon it. So I was working with I was lucky enough to work with a mechanical engineer, because I could come up with the optics,
Very cool.
in a design.
Interdisciplinary.
He needed to build it, because I can't build that kind of stuff.
Very cool.
And so he built this amazing, essentially like it's a big microscope with lasers, and we had all sorts of different lenses to, focus light properly and do all that kind of stuff. And so basically we were able to take, real live tissue, and instead of slicing it up, now we can shine light through it, different wavelengths of laser light, look at different parts, and now we can
Without destroying it.
Without destroying it. And now we can create essentially virtual histology, but now hasn't been corrupted in any way. And again, it's all digital, so it's quantifiable, right? Cool. And so that's why I loved it so much. And I like quantitative things more than qualitative, because I just find if you can put a number on something, you know more about it.
Yes,
that's just the mathematical side of my brain working. And so that's kind of where, I mean, people are still using this for also, actually a lot of neuroscience is being done with optical projection tomography at this point as well, to look at brain volumes in different, especially for things like autism spectrum disorder, Parkinson's.
Right.
So again, using mice, because this has to be really small sample sizes. This is not a technique that could ever be used on humans, but trying to understand morphology at these early stages and what leads to the downstream problems of these mutations.
So that was 13 years spent in a research lab. Let's talk a little bit about your time at Georgian. So what is your current role at Georgian? What do you teach? Do you do other things at Georgian as well?
Yes, so I'm a professor for the School of Health, Wellness, and Sciences, and I teach in 2 programs, so the Biotechnology program as well as the Pre-Health Sciences program. And I have another role at Georgian as well, where I'm the research lead for Health, Wellness, and Sciences. So this is where we report to the Research, Innovation, and Entrepreneurship Center, which is kind of a separate, I mean, it's part of Georgian, but it's not, has very little to do with teaching. It's this kind of separate, arm or entity of Georgian College. I think there are 4 research leads across the whole campus in different fields, right? So liberal arts, AI, and machine learning, and what's the other? I guess in more the kind of arts, like social sciences, that type of department. So I'm for Health, Wellness, and Sciences, research lead there. So that's how about a third of my time, I guess, is designated to kind of furthering research within the college. So this is a very different style of research from my past. This is community-based research, right? So what Georgian is really trying to do and is very, very good at, reaching out to community partners, saying, we have all of these resources. How can we help you advance your company, the community? Like, how can we help the city of Barrie even, or Grey Bruce, all these different areas, but very community-based, partner based research is what we're doing.
Which it's still at somewhat in its infancy, but will become very important to our students and a great opportunity
for sure
in the future.
So it's in its infancy, that's hard to say, for Health, Wellness, and Sciences, but other parts of the college are much more further down the road, especially when you think about the engineering departments. They've been working with community-based partners for a long time, but I really want Health, Wellness, and Sciences to be much a bigger part of this, because we have
so much to offer.
We have all the tools,
in skill and tools and
the students, and we've got all the students now that can do all this kind of work.
So, what is your approach or philosophy to teaching?
That's a great question. I really try my best to make things interactive, right? The last thing I ever want to do when I get into any type of a classroom is stand at the front and read information to the students. That's boring to me. I don't even want to do that on my end, let alone for them. So I really try to make things interactive, and I try to, the biggest thing that I really try to impress upon students is critical thinking, right? Can you solve a problem instead of trying to memorize a formula or something? Can you think about the logic of it, right? Can you think about, how do I get from point A to point B, right? And you can't skip steps, at least not at first, right? When you're first learning something. So trying to develop critical thinking skills by asking the right questions. I think that's important from our standpoint, right? Is to, if you don't ask questions in the right way, you can't get students to take the next step, right? Because if you think about it like a pyramid, right? If you start down here, you can't necessarily ask students to take 2 steps up the rung. You have to get them to critill think to get to the next step.
Right. There are no shortcuts.
Yeah, there's no shortcuts, but they have to be able to critical think to get to that next step on their own, hopefully. And then the next, you can't expect to jump 3 at once, right? So maybe 2 when you get some really good critical thinking skills. So I think my philosophy is to try to get students to have really good critical thinking skills and to kind of self-evaluate, what do I know about this and how can I synthesize
Right.
2 different areas or 3 different areas of information to answer a question.
Which I think
as
is,
opposed to like just, following
absolutely,
steps to get to an answer where you don't even know what you're doing anymore.
which I think is very appropriate and important for biotechnology, since biotechnology is using biology to solve a problem
Yep.
or to invent a product or a service. So
Yeah.
in order to be able to do that, thinking critically is absolutely essential.
Yeah, you can't just be memorizing things. That doesn't help.
So let me ask you, what aspect of biotech or even health teaching in these 2 programs do you find most rewarding?
Honestly, meeting all the students. I love being in the lab, especially, right? Because that's where it's very interactive, and I get to learn about all the students. They, tell me about what their goals are for the future. I try to find ways that I can help them achieve that. Most rewarding is definitely being in the lab with students, getting to know them, trying to see how I can help them, get to that next level.
Absolutely.
I would say.
So you talk about
interactive. Yeah.
next level and students. Are there any practices or habits that you've seen in really successful students in the biotech program?
First and foremost, I don't even care what program it is, attendance.
Yes.
I say this all the time, and it's always true. The students who are engaged, that come to class and come to the lab prepared, are the ones who do the best. And by like week 6 or 7, I can always tell who are going to be right at the top, because they're there, they're asking questions, trying to understand, and honestly, like showing up, like be there.
And not just be there, be present.
Be present, yeah.
Yeah.
Like if you're just going to sit on your phone the whole time while we're trying to have a discussion, you may as well not be there. Being present, yeah, like that is number 1. Just pre-planning for things, right? Coming to the lab understanding, at least, you may not always understand everything that you're supposed to do that day, and we'll always talk about things beforehand, but coming in with the notion at least of what the topic is, whether it's class, lab, lecture, whatever, coming in just already thinking about it so that you can kind of hit the ground running, right?
Right.
And showing up, just show up and be involved.
Along those lines, and back to when we were talking about what a day in the life of a researcher looks like, one thing I don't think people realize or appreciate is when you show up as a researcher or working in the biotech field, oftentimes you're not told what to do.
Yeah.
You might have a goal, a project, but nobody's telling you what to do day to day. You have to critically think about what experiment, what questions to ask.
Exactly.
So this is a very good skill to
So
develop.
this is why pre-planning for everything in life, and again, I think it's for all of life, but that's exactly it, right? I never showed up to the lab just like, I wonder what I'm going to do today.
Right.
That's not how you would ever, survive in a field like
That's
that.
right.
You have to already have a plan based on things you've already done. Okay, today I have to get X, Y, and Z done, and I'm going to stay here till I get X, Y, and Z done, even if it doesn't work. But I have to do that so that I can get to the next day where I can then do A, B, and C or something based on what happened the day before.
Right.
But if you're coming into anything with no plan, doesn't matter what job it is or what class it is or whatever, I mean, you're going to fall behind.
And in worst case scenario, best case scenario, you're planning days in advance. Worst case scenario, something has messed up in the lab, and now you are completely from scratch, and now you have to problem solve in real time.
Yes, yeah.
Right.
So
that happens.
just pre-planning and being ahead of the curve, right? Being ahead of, just coming prepared, right? I mean, over-prepared.
So let's talk about then the future of biotech. In your estimation, what are going to be some of the most important skills or knowledge bases for future biotech students 5-10 years from now, trying to project what that industry is going to look like? What do you think?
So this is a really great question. I'm sure that you're going to have lots of people talk about some of the obvious things like ChatGPT. So I'll stay away from that because it's also not my area of expertise. But one thing that I really see being what's going to be a big explosion going forward, I've already kind of mentioned this idea of systems biology. So everything we've done for the last, I was going to say decades, maybe even almost 100 years now, has been very much let's, like if I think about human health, right, and the healthcare field, right? So my background, we've always been taking this approach of, okay, let's see what happens when we mutate this one gene. How does this affect the organism?
Poking and prodding in very specific locations.
But that's very scientific because you have to have only one variable that you're changing in an experiment traditionally to have an answer, right? So you can't be mutating like 3 or 4 things and saying, now you don't know what went wrong, right? So that has always been the approach. But now we're at the point, if I use the heart as an example, since that's my background, there are hundreds of genes that are implicated in, for instance, valve formation that are either upregulated or downregulated and are dependent upon one another, right? This one goes up. Yes, exactly. Then this one goes down, and it has to go in a very specific sequence, but hundreds. So we know all these things from individual experiments, right? So now the idea is, and this is where I think AI is going to be a huge help, of course, is that we have all of this information. This has been the problem for the last 2 or 3, maybe even more decades. We are collecting so much information, we have no way to deal with it. So chip analysis, right? Looking at different, it doesn't matter if it's RNA, DNA, or protein levels that are going up and down. You get these chips of like tens of thousands of genes sometimes that are up and down regulated. No one ever knew what to do with it. Exactly. So what? So what?
You have the information.
So
what do you do with it?
We never knew what to do with all this information, and now and I think this is why all of a sudden we've seen such a huge explosion in AI, because this is what happens. You build up something, you need to have a way to solve that problem, and I think that what's going to happen now is that this AI approach to taking like terabytes, if not more, whatever's after tera, I don't even know, terabytes of data, and making connections between it. So systems biology seeks to look at the totality of, say, an organism. I've got all of these different things that are at interplay, and systems biology has been around for decades as well, right? But doing things manually, right? I watched lots of researchers trying to make these maps. This thing goes up, this thing goes down,
the genome, the proteome, the metabolome, all these things that are interconnected.
And you make these gigantic maps that would take years, right? Whereas I foresee in the future that taking minutes.
Yes, absolutely.
To then say, okay, so now when this thing affects this, which affects this, which affects this, you know what I mean? As opposed to looking at everything individually. So this is where, if someone, if I think about biotechnology from this kind of biological side, this is where you have to be a really well-rounded student, right? Don't just, I see a lot of students who maybe groan when they have to take some sort of gen ed or comm course or something like that. What I would always recommend is taking some other course or courses that make you a more well-rounded student. So for instance, when I was an undergrad, every time I had an elective, I would always take an English course. Because I loved it already, but I think it made me a better communicator. I learned how to write really well and communicate well. Now, technical writing versus creative writing, very different, but there's still the same idea. How can I communicate properly?
That's right.
So, for instance, if someone's out there thinking about, if I think about this field, having some sort of computer programming knowledge, or Georgian College even has a big AI program. Now you can start to combine, say, biotechnology with computer programming or AI to try and then get into this field, which I think it's going to be huge, right? If you start thinking about you keep hearing this on the news, and I've been hearing it for years and years when I was at Sick Kids Hospital, individualized medicine.
That's right. It just hasn't happened yet.
No, it has, but we're on the cusp, and I think it's going to happen in my lifetime. We're right there, right? And now with the advent of AI, individualized medicine is going to be the future, instead of just, if you think about, say, chemotherapy, it's basically like using a bazooka to kill an anthill.
That's right.
As opposed to, if this gene is the one that's mutated in this person with this type of cancer, what is the right cocktail of different things that we can give them, right? Or even genetic therapies are coming down the pipeline. So that is going to be huge. If I think about the healthcare sector, is this explosion of AI is going to help us with all of this data to then really truly make systems biology a huge field. Like I foresee that will be an actual avenue of study in college, university, higher level education.
So what I think I'm hearing you say is 2 things. The skill in the knowledge base is definitely AI, but also to use electives as an advantage. Use it
100%,
it to not just as a bird course because you want to
Yeah.
get good grades. Use it to pad your resume, your skill set, so that you become more attractive in the future too.
Think about
employers.
what will, whatever program you're coming out of that program with a whole bunch of other students.
That's right.
So how do you make yourself a little bit different, right? If you took all this, same gen ed courses that everyone else did, you're no different. So now it's just going to come down to marks. But if you want to, for instance, be a better communicator, which is really important in science because I saw a lot of scientists over my years you could be the most brilliant scientist in the world, but if you can't communicate your ideas clearly, nobody cares.
That's right.
Nobody cares if you can't present it, if you can't write about it properly. You actually see a lot of communication officers being hired for big labs because they don't want to write the papers and they'd rather hire someone to do that for them, right? Because why not? That person has that skill set. So if you can find another skill set to accompany, say, your science, your biotech diploma, you're only going to make yourself a better candidate for all sorts of jobs. And you can say that when you get to an interview, right? Or you can write that on your cover letter.
Right.
I took this course on communications, as an example. You have to diversify, I guess is the right word, right?
Yes,
to diversify.
Okay, as we wrap up, final words of wisdom. What sort of mindset or approach do you suggest or recommend that biotech students put themselves in as they go through a biotech program and then they're looking for a job or career at the end? What is the mindset or approach of a successful biotech student and then somebody who's looking for a career?
Yeah, that's. I think I have to reiterate what I said before. Being prepared beforehand. If you are just showing up to things with no idea about what's going on, we know it. I can tell. You know what I mean? And when it comes time for me to have to write you a recommendation when you are inevitably trying to find a job, it's going to be a much weaker recommendation than the student who I can tell has intelligent questions to ask, is prepared, is engaged, is trying their best. I mean, everyone's probably always trying their best, but you have to pre-plan everything in this life. And I think that the sooner that you start doing that, the way that you start to think about things further down the road, as opposed to just waking up and saying, what am I doing today? The better off you'll be, right? Because that's just not how, it doesn't matter if you're working in academia or in industry, that's just not how you would ever be successful. You can't be, coming from your back leg. You have to be moving forward at all times. So just being overly prepared, right? Planning ahead. And I really do think that applies to all life in general. But in terms of a career, we will see it as your professors and, inevitable jobs that you go to in the future. Your employers will see that as well. It's a really obvious trait. It's really easy to detect. It'll make you a better student. It'll make you a better employee. Just pre-plan, right? Just think ahead. Go the next step. It's easier said than done sometimes. The life of a student isn't always easy, but you have to start sometime, right? So start now. Yeah.
Thanks, Greg.
Thanks a lot. It was great.